Welcome to Unit 4. In this unit, we will continue our discussion of motor systems, specifically looking at the cerebellum and then later on also the basal ganglia. This one is going to be mostly the cerebellum, but I will make a few comments about both to get us started. So when you compare the cerebellum with the basal ganglia, it's going to maybe in the onset sound like some of their functions are overlapping, and that both of them have some way of modifying the motor signals that leave the brain to control the muscles. But if you look at them a little more closely, there is some distinctions. The cerebellum has to do with things like timing of motor activities and to progress from one muscle movement to another, particularly if you have a fast motion, like you're doing something quick like typing or any kind of quick kind of muscle progression from one activity to the next. It also helps to control how intense a contraction is, especially if the load that you're carrying or something like that changes, that it is helpful for that. Also, it helps to coordinate the agonists with the antagonists. So when your biceps are contracting and what are your triceps doing? Sometimes you want them to relax completely, but sometimes if you want to hold a joint steady or still, you want to contract both of them to some extent. So some of those things are a little different than the reflexes that we've talked about earlier. Also has some assistance in posture and equilibrium. So we looked a lot at posture and equilibrium before, having to do with our vestibular apparatus and some of our brainstem nuclei. But our cerebellum is going to have some assistance in modifying and improving on those activities. The basal ganglia has some planning activities. Well, cerebellum in some sense does too, but in the basal ganglia seems to have maybe a different way of doing this, but it helps to plan and control complex movement patterns. It has another one of those functions to maintain the right amount of intensity of different movements too, and the direction of movement, and also with sequencing of movements to achieve a function that you want to do. So that sentence sounds a little bit like what the cerebellum we just said. So yeah, there is a little bit of things that are a little overlapping, but their circuitry are different. Let's look at our cerebellar anatomy a little bit. Perhaps you looked at this in neuroanatomy already, but the cerebellum is said to have an anterior lobe and a posterior lobe. With an upright human, anterior lobe is actually kind of superior and posterior is sort of inferior, but if you think in terms of a animal that's horizontal, anterior and posterior actually makes more sense if you think of it that way. And tucked underneath here we have another little lobe called the flocculonodular lobe. We'll make a few comments about that one, but it's the smallest of the three lobes. From a functional point of view, it is helpful to divide up the cerebellum into zones. And this is a posterior view of the cerebellum, and what they've done in this illustration was sort of uncurled it. So right now we see the cerebellum sort of curled. If we were to flatten it out, it would look something like this, so that we can see that here's our anterior lobe and our posterior lobe and our little flocculonodular lobe down here, and we see we have color-coded some zones. So these are the zones that we're going to care about, and these zones span both the anterior and posterior lobes. Along the center we have the vermis, and this has to do with helping to control the axial body, the neck, shoulders, and hips. If you want to think about those parts of your body and the skeleton and the muscles that control that parts of the skeleton, this is sort of like your background framework. This is your core that your extremities then have to depend upon to be able to do their job. You know, you have this axial activity, and our vermis is useful for that. In the blue area, which is the intermediate zones of the two hemispheres, hemispheres being on either side of the vermis, by the way, the intermediate zone's job is to coordinate the distal limbs, especially hands and feet, but beyond where the vermis did. And then the lateral zones out here have more to do with the sequencing of movements and with the coordination with the cerebral cortex, of course, but helping to sequence movements. And this is not specific to any particular part of the body. The cerebellum is attached to the brainstem. Remember, the cerebellum itself is not considered part of the brainstem, but it is sticking out the back of it. So here, once again, is our pons, and here's our medulla. And if you were to cut the cerebellum right down the vermis, and you look at it from the medial side, this is what it would look like. You would see that it has white matter inside it, and the white matter looks like a tree, so they call that the arbor vitae. And then on the surface you have some gray matter, and that gray matter over in the where I'm pointing is in the cortex of the cerebellum. So those are the folia of the cerebellar cortex. Not shown here, maybe they didn't quite get right in the section where they would be, but there are some deep cerebellar nuclei as well that exist. Again, we can't see them right now. Here is our fourth ventricle, so we can see where we're at, and there's the choroid plexus of the fourth ventricle. And this is the cut end of the other half of the cerebellum that was removed to see that medial aspect of the cerebellum on the other side. And these are those cerebellar peduncles that connect the cerebellum to basically the brain stem. Do we have a topographical representation in the cerebellum? Well, yes and no. If you're asking about the vermis and the intermediate zones of the cerebellum, then yes, we do have a homunculus of sorts. Although the homunculus is a little odd in that the anterior lobe has a different homunculus than the posterior lobe. However, in both cases, along the vermis is where you're going to see the axial parts of the body once again, and then the hands and feet are going to be controlled largely by those intermediate zones. And there apparently isn't any homunculus in the lateral zones, that there doesn't seem to be a topographical representation of the body in those most lateral parts of the cerebellum. Now, what's interesting to note is that the cerebellum does not directly initiate any motor contractions. So in other words, if you were to take an electrode and stimulate the cerebellum somewhere, you would not get a muscle to contract somewhere in the body. That's very different than what we see with our motor cortex. If you were to stimulate some part of the motor cortex, like the precentral gyrus somewhere, you would get muscles to twitch someplace. The cerebellum does not directly initiate any motor activity. However, what it does is it receives all kinds of input from a variety of sources and then sends output back to the cortex in some places to modify the plan or modify the work in progress, the activity in progress. Once again, it does not initiate its own motor control. So cerebellum does receive continuous, up-to-date information about what the intended motion that your motor cortex wants to do. So it finds out what the plan is from the motor cortex. And it also, at the same time, receives all kinds of sensory information from the periphery, like proprioception and other things. So it knows where the body is in space and what is happening in a moment-to-moment basis, and compares the two so that, well, what did you want to do and what is actually happening, and compares them and makes adjustments. Not only is it modifying the activity as it's progressing, but it has a little bit of a planning portion to this as well. So that once you know where you're at with the current movement, it can help to smooth the progression. That's why it's so important when you have fast movements. The cerebellum helps you to transition to that next thing more effectively. Cerebellum is important also in motor learning, in that it helps you to learn from mistakes. So if you do a layup of the basketball and you try to get it into the net and it misses wildly, cerebellum is going to be very helpful to help to make adjustments so that your next attempt may be a little closer, and keep doing that until the motion is improved. So let's take a look at all these afferent pathways that go into the cerebellum. Okay, so this is afferent into the cerebellum. One is called the corticopontocerebellar pathway, and the name tells you where it's going. It starts in the cortex, particularly the parts of the cortex that have motor function, promotor, premotor, and yeah, remember the somatosensory cortex had some motor function? Okay, all of those areas. Cortico, and then it relays in the pons, some nuclei in the pons, pontine nuclei, and then back to the cerebellum. Corticopontocerebellum. And where in the cerebellum? Mainly in those lateral zones. So those are afferent pathways into the lateral zones. You also have olivocerebellar pathways into the cerebellum, going from the motor cortex, also basal ganglia, also some reticular formation, and even the spinal cord. And that will then relay in the inferior olive, there's the olivo part, and then goes to all parts of the cerebellum, olivocerebellar. We will learn that these are the origin of one particular type of afferent that comes into the cerebellum, and that's the only source of those particular kind of fibers. We'll get to that. Vestibulocerebellar, as the name implies, comes from the vestibular apparatus and goes to the cerebellum. So vestibular apparatus and heads to the flocculonodular lobe or vestigial nucleus, which is one of our deeper nuclei within the cerebellum. So it could have some fibers going directly from the vestibular apparatus to this part of the cerebellum for balance type things, or could be relayed by one of those vestibular nuclei. You also have reticulocerebellar pathways, going from the reticular formation, which we learned about a little bit in our earlier unit, and then heads to the cerebellum. And these tend to go to the vermis. A few other afferent pathways, since I didn't have enough space on the previous slide. We have our dorsal spinocerebellar tract. The dorsal spinocerebellar tract contains a lot of our peripheral mechanoreceptors, things like muscle spindles, Golgi tendon organs, various tactile receptors, receptors in our joints, and a lot of this then informs our vermis and our intermediate lobes, and they tend to be ipsilateral tracts, by the way, but anyway, dorsal spinocerebellar tract, often referred to as unconscious proprioception, even though a lot of the same information goes to our sensory cortex for our conscious awareness of things, but our cerebellum gets a copy of this information as well, and that's the part that is unconscious. We aren't aware of this information landing in our cerebellum. Now the coolest pathway, afferent pathway, I think, is the ventral spinocerebellar tract. And what happens with this one is that, remember that in our spinal cord, let's just make a real quick little spinal cord right here. And sorry if it's a little ugly. Make a little gray, it's terrible. Make a little gray matter. All right, this person has problems. But anyway, that we had a anterior motor neuron, there's our alpha motor neuron leaving, and we had learned earlier that there's lots of other tracts that impinge on that, right? Some coming from the other side or what have you. All kinds of interneurons, okay, all kinds of things that are impinging on that anterior motor neuron. Now, whether this neuron fires or not, it's going to depend on the summation of all those different influences. Well, how does the cerebellum know way up in the brain how that went? How that summation actually came out? Did this neuron actually fire or didn't it? Well, there is sensory tracts via the ventral spinocerebellar tract that go up from that neuron to the cerebellum to let the cerebellum know what happened. They call this the efferent's copy. Okay, so the signals that actually arrive at the anterior motor neuron is going to excite this tract to let the cerebellum know what the pattern of firing of those motor neurons are. Now, as you might expect with these afferent pathways to the cerebellum, they have to be fast. If your job is to modify movements in progress and to make quick adjustments to things to smoothly progress to the next movement, you got to have fast fibers. So these are those A-alpha 120 meter per second speed types of fibers. Now, we didn't see that the deep cerebellar nuclei in a previous picture, but we can see it in this one. Here they just highlighted those deep nuclei. These are gray matter kind of scattered within the cerebellum. And I'm going to describe three of them. Dentate nucleus is shaped sort of like a tooth, I guess. And interposed nucleus. Now, technically the interposed nucleus is made up of two smaller nuclei called globose and emboliform. I don't care about that for this class. We'll just take them together and refer to them as interposed. And then we have our fastigial nucleus. And our fastigial nucleus is the green one. Okay? Now, all cerebellar nuclei that we just listed, all of them, receive input from two sources. Either direct sensory afferents, remember that list of afferents we just went over? Direct sensory afferents from outside the cerebellum into these deep cerebellar nuclei, and those afferents coming in direct to those nuclei are excitatory. And then we'll see that there is an indirect pathway where some of these afferents go somewhere else, mainly the cortex, the cerebellar cortex, and then find their way back to those deep cerebellar nuclei. And when you take the roundabout route to get back to those deep nuclei, they tend to be inhibitory. And we'll see how that is really important in nuclear and cerebellar function. Outputs out of the cerebellum always start in one of those deep cerebellar nuclei. So if there's any efferent leaving the cerebellum going somewhere else, it's going to be coming directly from one of those deep nuclei. So what we're going to look at is those three nuclei again and discuss the pathways. The dentate nucleus, let's look at that one first. The dentate nucleus receives input from the lateral zone of the cerebellum, and then after it gets to the dentate nucleus, then it goes out again through the thalamus and then through the cerebral cortex. Now remember, the lateral zone had to do with not any particular part of the body necessarily, but is a general area, right? We didn't see homunculus in those lateral zones. However, these coordinate sequences of motor activities that are initiated by the cortex. So if you remember our corticopontocerebellar pathway that goes to those lateral zones, that's the plan from the cortex, and this is going to be going through this process through the dentate nucleus back to the cortex to coordinate those sequences of activities. Interposed nucleus. These receive input from the intermediate zones and through the interposed nucleus, and then from out of there goes to pretty much all of the motor areas you can imagine. Motor cortex, basal ganglia, etc. Lots of places. And this has to do a lot with those distal limb activities, coordinating those distal limbs, especially timing the coordination when your agonist and antagonist muscles are contracting. Fastigial nucleus. This receives input from the central part, the vermis, and then after it leaves the fastigial nucleus, it goes out to the medulla and the pons. And remember, the medulla and the pons had a lot of those systems that had to do with postural muscle control, and that's right, because isn't that what the vermis is for? So it functions in association with the vestibular system and the reticular formation, for that matter, probably as well, to help with those postural and equilibrium kinds of control. If we zoom in on our cerebellum a little bit and look at our histology, here is a low magnification view, and we can see the arbor vitae. I know it's normally white, but in this picture it's dyed, so it's kind of this purple color. And then we can see the granular cell layer, and then the Purkinje layer is too thin to see at this magnification. And then you have the outer layer of the molecular layer in the lighter pink. If you zoomed in on a little piece of this area, it would look something like this. So here's that light pink molecular layer. This is a Purkinje cell. They're really conspicuous because they have huge cell bodies. And then deep to that you have your granular cell. Here's an artist's rendition of a real simplified view of that, where you have molecular cells, Purkinje cells, and then granular cell layers. And then under here you can't see them, but below that will be the deep cerebellar nuclei. So let's take a look at the circuitry a little bit. Now we have two fiber types that are of particular note, of particular importance here in the cerebellum. We have climbing fibers and we have mossy fibers. Remember how I said the olivocerebellar tract had some special fibers? Well, those are climbing fibers. So the inferior olives is the only source of these climbing fibers. So we have a climbing fiber illustrated here. Here's a blue cell coming in. And one branch will go to a deep cerebellar nucleus, and another branch will go more superficial towards the cortex. Mossy fibers, those are all the other afferents. Whether they're corticopontocerebellar fibers or they're spinocerebellar tract fibers, whatever they are, if they're not from the inferior olive, they're a mossy fiber. And there's an illustrated one here. Once again, the pattern's kind of similar though. It has a branch that goes to a deep cerebellar nucleus. Remember, direct control to the deep cerebellar nucleus. And then you have another branch that comes off to the surface someplace and goes elsewhere. Those are mossy fibers. The Purkinje cells are the red ones, and we'll describe those a little bit differently. This is a more simplified diagram than the previous one, and this is a little bit more helpful. Okay, so once again, we have a deep cerebellar nucleus. That's the blue dot right there, deep nuclear cell, and there's its axon leaving. Remember, all exits out of the cerebellum come from a deep nuclear area. Its axons are leaving here. And we have mossy fibers coming in, and we have input from the inferior olive, which are climbing fibers. And we said in an earlier slide that our deep cerebellar nuclei get direct innervation from sensory input, and that's what we see here. And they tend to be excitatory. That's why there's plus signs. That's excitatory. And then we said there's indirect inhibition. So let's take a look at that idea. Our mossy fiber, for example, will come up here, and it will synapse on another population of cells called granule cells, hence the granular cell layer. And then the cell's axons will come up here, and the axons will then move sideways along the cortex of the cerebellum. And they, in turn, will synapse with these black cells here. This is a Purkinje cell. And that Purkinje cell then synapses with the deep cerebellar nucleus, and that is inhibitory. So this is a direct pathway that's excitatory, and an indirect pathway going around the whole way this way through the Purkinje cell is inhibitory. The climbing fibers have a similar process, except they aren't using the granular cells in the same way apparently, but they do have a direct input into the deep nucleus and then a roundabout way using the Purkinje cells to cause inhibition. So how is this doing anything? What is this? Why do we go bother to look at the circuitry? Because it has a lot to say about how our cerebellum does its thing. It's sort of like a turn on and turn off signal for coordinating muscle activity. So essentially, what the turn on signal does is to accentuate a voluntary initiation of a movement. For instance, signals from the corticopontocerebellar tract, which would be mossy fibers, synapse directly with those deep nuclear cells, and that will then send output that accentuates that activity. So let's just back up for that in a minute. So this is corticopontocerebellar mossy fiber. That will accentuate this output, and that will be synergistic with or excitatory with an onset of movement. The turn-off signal, however, functions to help time when that muscle relaxes. So mossy fibers also synapse with those granular cells with those tiny little projections, which we just said over here, and goes around the other way, activates the Purkinje cells to inhibit, and that will turn it back off again. So this will stop or help to suppress the contraction. So you have a timing function here, turn on, then turn off. Now, what about the pace of that turn on, turn off signal? That's the job of the climbing fibers. Okay, here's the turn on this way, turn off going around the long, slower way to turn it back off again. Now, the timing of this turn off is going to be modified by the climbing fibers. So here's the interesting thing. If there is a poor match between what your intended movement was and what the actual movement was, the climbing fibers are the most active. When there's a mismatch, when the cerebellum compares what you wanted to do and what actually happened, and if there's a mismatch, these climbing fibers start firing a lot more. And that changes the pattern of firing of the turn off, or the timing of the turn off, and help to then improve the outcome of the next attempt at doing this activity. In this way, the cerebellum learns how to time a motor skill better. Now, this isn't instantaneous. Clearly, you try something new and then it fails, and then you try it the second time, you weren't perfect, but it does continue to make adjustments as long as the what you wanted to do and what actually happened are different. So to put together all of this together, we have then three different descriptions of the cerebellar circuitry. We'll call the vestibulocerebellum function as the parts of the cerebellum that include the flocculonodular lobes and the vermis, and that deep cerebellar nucleus that is most associated with those parts of the cerebellum was the fastigial nucleus. And this has a lot to do with the modifying equilibrium, giving you that core stabilization for all that movement that might be happening with that core in the center, and might anticipate corrections necessary for when you're doing things with these limbs to maintain equilibrium and that status quo. The spinocerebellum is associated with the intermediate lobes, and the deep cerebellar nucleus that is most associated with that is the interposed nucleus. And the intermediate lobes, remember, had to do with distal limbs, and helps to, again, make sure the movements of the distal limbs are matching what you want to do, and helps prevent overshoot of motion. What do I mean by that? If I was trying to point at something, let's say I'm pointing at my camera. If I point at the camera and I miss, it's an overshoot. And oops, I overshot it again. Oop, now I got it. Okay, so preventing that overshoot, the cerebellum can be very helpful with that. The cerebrocerebellum, this is associated with the lateral zones part of the cerebellum. The deep cerebellar nucleus associated with the cerebrocerebellum are the dentate nuclei. And this is, again, working with those lateral zones for that planning and sequencing and timing of multiple groups of muscles and things like that. Also, it says helps to time extramotor predictive functions. What do they mean by that? If I'm walking and at some point I'm going to hit something, a wall or a tree, well, you can plan when to turn around. Yeah, that's a part of the function of the cerebellum, is to help you to predict when that contact would happen and prevent it. It's also sometimes helpful to understand what the cerebellum does is by describing what it looks like when the cerebellum doesn't work correctly. For instance, with cerebellar dysfunction, you might have an intention tremor. And what that means is that, well, it's a little bit like that overshoot idea. I might intend to point directly at that camera, right? However, if I go and beyond it and overshoot it, then I will try to correct, and then I'd hypercorrect, and then I'd hypercorrect again and again. So you do this. Every time you try to point at something, it's hard to pinpoint where you want your finger to go. That's an intention tremor. When you are doing your intended movement, that is when the tremor happens. That's in distinction with what happens with some basal ganglia dysfunction, where you have a resting tremor, like with Parkinson's, for example, where the tremor's at rest, and actually the tremor's less when you do your intended movement. So it's really the opposite with the cerebellum. Okay, dysmetria, an ability to predict how far a movement will go. You're misjudging how far movement is going to happen. Ataxia is a term, generally just ataxia means a bad gait or a disturbed gait, but there is a type of ataxia you see with cerebellum, and that will have a certain pattern that we'll see in a little video I'll show you. Dysdiadochokinesia, there's a big word. That is an inability to form rapid alternating movements, like for example doing something like this. I'm slapping my hand back and forth as fast as I can, and if you can't do that, that might indicate a cerebellar dysfunction. Dysarthria is a failure of progression in talking, which sometimes I think I have when I have to keep repeating myself, but tripping the tongue kind of sort of failure of progression in talking. Hypotonia is a lack of muscle tone because the cerebellum facilitates some of those motor output signals, the turn on parts anyway. If they're not doing their part, then perhaps overall the tone of muscles is going to be decreased. So we see hypotonia sometimes with this cerebellum. The eyes also would be affected too. So if you examine the eyes of a person with cerebellar dysfunction, you might have a nystagmus, which is a tremor of the eyes. So it's similar to like an intention tremor, but with the eyes. And what's really cool about them is that your eyes will flick, say, to the left, and then they'll return back to the right in that tremor. That the one direction will be fast and returning will be slow. And whichever side is the fast phase, that's the side of the cerebellum that is most damaged. Now in this little video we have some cerebellar function tests that are illustrated, and I'll ask you a few examples about that. But for instance, you have that gait abnormality, also a test to demonstrate rapid alternating movements. There's also something called the finger-to-nose test. And the finger-to-nose test is similar to the heel-to-shin test, except the finger-to-nose test has to do with the testing the upper extremity joints and your ability for your cerebellum to coordinate that. And the heel-to-shin test is not illustrated in the video, but is a similar sort of thing with your lower extremity, where you trace your heel along your shin up and down your leg and to see if you can coordinate that activity. Same basic idea, but just using the different extremity. The function tests that are shown in that video also discusses the Romberg test, which sometimes is done with a cerebellar evaluation, but really whether or not you have your eye, you'll see what it does, but whether or not you have your eyes closed or not, if you have a cerebellar dysfunction, you're going to be positive anyway. So really the cerebellar function test has more to do with vestibular apparatus or your proprioception coming up your dorsal columns. It's more of a test for that than it is for the cerebellum. Anyway, when you get a chance to look at those tests, you'll understand what I mean. Okay, that is the end of the cerebellum. We will tackle the basal ganglia next.